Okay, as we're all getting settled in, it's truly an honor for me to be able to introduce our next speaker. Dr. Bill Kaelin is a Howard Hughes medical investigator and a full professor of medicine at the Dana-Farber Cancer Institute and the Harvard Cancer Center. Over the course of his career, he's published really seminal work in many of the most critical cancer pathways, including von Hippel-Lindau or VHL, hypoxia-inducible factor, tuberous sclerosis, and mutant IDH or isocitrate dehydrogenase, which we'll hear about today. He's also received probably every prestigious cancer award there is, including the Lasker Award, the Science of Oncology Award from ASCO, and he was also elected to the National Academy of Science in 2010. But for my contemporaries, Dr. Kaelin's really known as an outstanding mentor. Many of his former trainees have gone on to start really outstanding labs at Harvard and elsewhere. But he's also been an integral part of training people who are not in his lab. So he's been a key coordinator of the AACR Molecular Oncology Workshop, which I attended, where people who did not join his lab were able to benefit from his mentorship as well as other leaders in the field. So without further delay, Dr. Kaelin, thank you very much.
Thank you for that overly generous and kind introduction. It's really very nice to be here. And I understand we have a mixed audience, so I will apologize in advance for my jargon-laden scientific talk that I was asked to give. But the take-home message is there are literally thousands and thousands of investigators around the world working on the cancer problem, including 15 very talented people in my lab. Unfortunately, many of them are far more gifted than I am. So I'm just the spokesperson, and I hope you get some flavor of the work that we are doing in our laboratory, and then multiply that by thousands, and you get a sense of the effort that's being made here. So here are my obligatory disclosure slides. And I'm going to tell you a little bit about our work on cancers that have these so-called IDH mutations, and this was largely the work of Sam McBrayer, a postdoctoral fellow in my lab, and Diana Shee, who's a medical student working in the laboratory. And here's just a partial list of the collaborators who have helped us. And I show this in part because you sometimes hear that scientists don't work together, and there's nothing further from the truth. We all rely on collaborations that we form around the country and around the world. So a number of years ago, it was determined that some cancers have mutations in one of two genes called IDH1 or IDH2 for short. I show in red here some of the cancers where one has found IDH mutations, and of course I'm here because of the link between IDH mutations and cholangiocarcinoma. And these mutations, we now know, are unusual in the sense that they lead cells to accumulate a little chemical that scientists refer to as R2HG for short. But this chemical accumulates to very high levels, and the problem is there are a number of proteins in the cell, and in particular enzymes, that normally would use a highly related chemical, but when the cell has too much of this chemical, it tends to interfere with these enzymes, and this leads to certain forms of cancer. Now, among these enzymes, there are enzymes that we know are actually the enzymes that, when altered, are causing the cancer. And so, for example, in leukemia, we know that an enzyme called TET2 is really one of the critical targets of this 2H G I referred to a moment ago. But we also know there are a lot of other enzymes that are being altered by the 2HG that are simply innocent bystanders. They're probably not causing the cancer, but nonetheless their function is being altered. So we were intrigued by these IDH mutations in cancer, and so Julie Lossman in the lab began by studying the role of IDH mutations in leukemia. And what she needed was a laboratory assay where she could monitor the ability of these IDH mutations to promote cancer-like behavior, such as abnormal proliferation. So here, she's taken a leukemic cell line called TF1 cells that, despite being leukemic, grow very poorly in the laboratory unless you give them a fertilizer scientists refer to as cytokines. But she discovered that if she introduced into this leukemic cell line shown in blue, an altered version of IDH1 that had been seen in a variety of patients with leukemia. Now these cells acquired the ability to grow even if they weren't given that fertilizer or the cytokine compared to the control cells. And then she asked, could this really all be about this little chemical, this 2HG? Is 2HG really the mediator here? And the answer was yes, because we collaborated with a scientist actually here in Utah, Ryan Looper, who's a very clever chemist. You can't just give cells this 2HG because it won't get into the cell, but he made a version of the 2HG that can actually enter cells and get trapped. And so when Julie gave this version of the 2HG to these TF1 cells, now they acquired the ability to proliferate, just like she had seen in the previous slide with the IDH mutation itself. And again, for the specialists in the room, 2HG comes in two flavors, one the chemists call the R form and one the chemists call the S form. And it's the S form specifically that's causing cancer. And when Julie tested the other form, she saw the other form doesn't do this. So this is really exquisitely specific. It's this one altered version of this one teeny little chemical that's causing the problem here and causing these cells to proliferate abnormally. So, unfortunately, there are a lot of naysayers in science and in clinical medicine, and almost immediately people started doing hand-waving arguments for why the effects of this bad 2HG would not be reversible. And if that was true, then there'd be no sense in developing drugs that would prevent the production of 2HG. So Julie decided to try to test that in the laboratory, and she did this a couple ways. I just told you a moment ago we can give the cells 2HG and they'll start proliferating. And there's a little trick here, which is you have to give the 2HG for 20 passages in cell culture. So in dark blue is the result I just showed you, where she's given the cells the R2HG, and now the cells can proliferate. But if she could give the cells R2HG, she could also take it away. And that's what's shown here, where she gave the cells the R2HG for 20 passages, but then for varying amounts of time, then took it away. And what you saw was that the cells reverted to normal, such that eventually they looked like the cells that had never seen the R2HG. So that was encouraging. We also collaborated with Agios Pharmaceuticals, and I have a small financial conflict of interest here. Agios had developed a tool compound you're going to hear more about, and the cousins of this tool compound, that will bind to the mutant IDH and prevent it from producing the 2HG. So with this tool compound, we can now ask it in a different way whether cells have an ongoing requirement for 2HG to proliferate. And so here in the box up here, we're measuring the amounts of 2HG in cells. So WT means wild type or normal cells, and they have very low levels of 2HG. If you put in the IDH1 mutation, however, now you see really industrial amounts of the R2HG. But when Julie treated these cells with the Agios inhibitor, she saw that there was about a 10-fold reduction in the amount of 2HG. And then she asked whether in the system I just showed you a moment ago, did this actually translate into impaired proliferation? And the answer was yes, because in dark or royal blue, you see the result I showed you earlier, where the cells that have this R132H IDH1 mutant can now proliferate in the absence of the fertilizer, the cytokine. But if you take these cells and now treat them with the Agios inhibitor, now they stop growing. Now that's extremely encouraging, and it's consistent with the idea that this drug is working by blocking 2HG production. But with any chemical, we always worry maybe there are some other activities of this chemical. Maybe this is really a rat poison. We don't need any more rat poisons, even if it does block 2HG. But Julie realized there's a way to ask whether this is really specific to the R2HG, because if she takes cells and instead of having the mutant IDH, she's giving them that version of 2HG that can enter into the cell and get trapped into the cell, these cells are no longer affected by the Agios compound, presumably because they're already getting what they wanted. So if you're already giving the cells 2HG, now the cells are insensitive to a drug that prevents the production of 2HG. So this is another criteria. This is a very specific drug that is capable of blocking 2HG production. For technical reasons, the Agios compound that inhibits IDH2 went into the clinic before the Agios compound that inhibits IDH1. But suffice it to say that even in the early phase 1 sort of dose-finding studies in these patients with acute leukemia who had IDH2 mutations, some of the patients went on to have complete remissions, the so-called CRs, and some of them even went on to consolidative bone marrow transplants. So this is the kind of thing you would like to see, where some patients now are going into complete remissions. I think now that the dust is settled, it's about 30% or so of patients with acute leukemia that have IDH mutations have these really very dramatic responses to these drugs. So then we turned our attention to brain tumors that have IDH mutations. And once again, we realized we needed a model we could study in the laboratory to monitor the effects of the mutant IDH and the 2-HG, and then the drugs that prevent 2-HG production. So here, Sungwoo Lee in the lab and Pepi Koivunen took immortalized human astrocytes, which are a type of brain cell, and introduced into those brain cells either the normal version of IDH1 or this tumor-associated version of IDH1 we referred to a moment ago, this R132H mutant. And for good measure, he also introduced into these cells a version of IDH1 which has simply been inactivated, so it's sort of an inert control. But if he put in the mutant IDH1, these cells can now grow in a gelatin-like matrix and form what scientists refer to as soft agar colonies. And this is another surrogate for badness, that these cells are on their way to being cancerous because now they can grow in the soft agar matrix. Nonetheless, these cells won't form tumors if you inject them into, for example, immunocompromised mice. To do that, you have to add another mutation. And we know in these brain tumors that have IDH mutations, there are almost always additional cooperating mutations in other genes that are causing the cancer. And so one combination we knew of was an IDH1 mutation combined with a mutation in another gene, and I apologize for the jargon, called PIK3R1. But if you have the astrocytes now with a mutation in this gene and a mutation in this gene, now if those cells are implanted into immunocompromised mice, they will form tumors that we can monitor as shown here. And so this looks, again, like the mutant IDH really is a bad actor here that is causing these brain tumors, especially when combined with other genetic events. The bad news was, however, if we treated those tumor-bearing animals with the Agios inhibitor or the equivalent thereof, in stark contrast to what we had seen in IDH mutant leukemia models, these tumors, much to our surprise and dismay, just kept growing. So here we're looking at the growth rate of the tumors in red treated with the IDH inhibitor versus in blue the control. And this is despite the fact that the drug was doing exactly what it was supposed to be doing, because when we measured 2-HG in the tumors, as we had hoped, with the Agios drug, we could lower the levels of 2-HG. I see now one of the labels was reversed, but the point is the same. So the drug is doing what it's supposed to be doing. So we might need other ways of killing or controlling these brain tumors that have IDH mutations other than the more direct approach of just cutting off the supply of 2-HG. And so here the idea would be maybe we should leave the 2-HG levels very high, but we should exploit the vulnerabilities that are created in a cell when they have too much 2-HG. And to a scientist, this would be referred to as sort of a so-called synthetic lethal approach. So we're going to find the vulnerabilities that are created when cells have too much 2-HG rather than simply taking the 2-HG away, because as I just told you, at least in the laboratory models, the latter doesn't seem to be very effective. So Sam McBrayer in the laboratory is a very talented postdoc, and he had read a paper that had already been published where investigators at Duke had generated cell lines that were identical except for whether they had normal IDH or mutant IDH. And then the investigators had looked for various chemicals, specifically metabolites, and asked whether there were metabolites that went up or down in the cells that had too much 2-HG. And so, for example, here's in yellow showing that the cells that have mutant IDH have too much 2-HG, but that's expected. So yellow is up and blue is down. But the cells with the mutant IDH also had low levels of these metabolites shown here. And I realize these are mouthfuls, but here are the acronyms that scientists use to refer to these chemicals. And Sam's very talented, and he realized that these three chemicals that were missing in the mutant IDH cells are all produced by one of two enzymes that are called BCATs, for short, branched-chain amino acid transaminases. And these enzymes are another example of enzymes that require 2-oxoglutarate to function. And so again, I've already told you that 2-HG can often compete with or interfere with 2-OG. And so that beget the following simple idea, that the R2-HG in the IDH mutant brain tumors were inhibiting these branched-chain amino transaminases, these BCATs, and that's why you were missing KIC, KMB, and KB. So Sam actually developed biochemical assays where he could monitor BCAT activity in a test tube, and then he could add the 2-HG and ask whether the 2-HG was an inhibitor. And so shown here in red, green, and purple, he's adding in more 2-HG into the enzymatic reaction in a test tube. And these are tumor-relevant concentrations of 2-HG. And he could see that indeed, as he hypothesized, 2-HG can inhibit the BCAT reaction across a range of 2-OG concentrations. And this was specific because I told you 2-HG comes in another flavor, the S2-HG. The S2-HG doesn't do this. So the tumor-causing 2-HG inhibits the BCATs, and the control, the S2-HG, does not. And getting back to collaborations, we've been collaborating with a group at Penn State that had solved the structure of the BCAT1 protein and the BCAT2 protein, and they've helped us understand exactly how the 2-HG fits into a pocket in this protein and inactivates it. Okay, so what can we do about this? So Sam and Diana created, again, matched cell lines that are going to be identical, except they're either going to have wild type or normal IDH1, or they're going to have this tumor-associated IDH1 mutant. As expected, if the cells have the tumor-associated IDH1 mutant, they make too much 2-HG, whether they're grown on a plastic dish or whether they're implanted into the brain of an animal. And then the hypothesis is in these cells, this BCAT1, BCAT2 reaction should be inhibited. And so to see if this really took place in cells and not just in a test tube, Sam obtained a so-called isotope version of leucine that you can monitor, you can now unambiguously monitor the conversion of this leucine into glutamate by BCAT1. And as expected, if you monitor the rate of conversion of leucine to glutamate in the cells that have the mutant IDH1, the rate of conversion is substantially diminished. And you can do this with the cell membrane permeable version of R2-HG as well. So 2-HG is actually sufficient to suppress BCAT1 and BCAT2 in cells. Is it also necessary? So here again, we could exploit this wonderful compound from Agios, where we can shut down 2-HG production. So that's shown here. So if you take the mutant IDH1 cells that have high levels of 2-HG and you give them the Agios inhibitor, 2-HG is almost normalized. And now if you repeat those Flux studies in the cells that previously had suppressed BCAT1 activity, if you treat them with the Agios inhibitor, now you normalize the flux through BCAT1 and BCAT2. So 2-HG is both necessary and sufficient in this model to suppress BCAT1 and BCAT2 enzymatic activity. So why do we care about this? So cells really need and like a amino acid called glutamate. It turns out you can make glutamate a couple of different ways. One way I just told you is that you can start with 2-oxyglutarate or 2-OG, and then BCAT1 and 2 will convert the 2-OG to glutamate. But I've also told you that in the cells with an IDH mutation and too much 2-HG, they have a problem because BCAT1 is now inhibited. But cells have a fallback, and one of the fallbacks is to take another amino acid called glutamine and convert it to glutamate. And that's carried out by an enzyme called glutaminase. And fortunately, there's a company called Calithera that's made a drug that's in clinical trials now called CB839 that's going to inhibit glutaminase. So this is a perfect setup for what I told you about earlier, this concept of synthetic lethality. So now the cancer cells have lost this arm, so they should be hyperdependent on this arm. Now, it turns out this had already been suggested by others, but not for this reason. There were other reasons people were hunting for vulnerabilities, and a group had stumbled upon the idea that if you inhibited glutaminase in IDH mutant cells, they were sensitive, and that's shown here on the right, where in the turquoise, the cells with the IDH mutation are being treated with an agent that will eliminate glutaminase compared to the control in gold. But a scientist would recognize there were a couple problems with this experiment, not the least of which is this anti-proliferative effect is quite modest and is almost, is unlikely to translate into a meaningful clinical benefit. But we realized if our model was right, these investigators might have wildly underestimated the utility of this approach because most scientists, including I was one of these people too at one point, don't really think about the media they're buying from the vendors. And if you're not careful, the media you're buying from vendors to grow your cells on plastic dishes contain, really industrial amounts of a chemical called pyruvate and industrial amounts of a chemical called glutamate. And I'm using the word industrial because the amounts of these chemicals are orders of magnitude higher than would ever be seen in the body. And if our model was right, a good way to obliterate the dependence on glutaminase would be to just provide the cells with tons of glutamate and tons of pyruvate. And so Sam, being very smart, went back and made media that looked more like this rather than media that you buy from various vendors. And when he did this, the effect was so large, he now had to switch to a log scale. So some people say, wait a minute, Bill, this doesn't look that impressive. Well, it doesn't look that impressive because now we're on a log scale where this goes from 1 to 10 to 100. And that's the only way he could show easily in green the benefits of using a glutaminase inhibitor. And he also realized that if he was completely on mechanism, if he treated the cells with the Agios glutaminase inhibitor and removed the 2-HG, now the glutaminase inhibitor treatment would be the same whether you had an IDH mutation or you were IDH wild type, because again, by removing the 2-HG, these curves should now collapse, and that's what he saw. And then finally, to make the point about the importance of how much glutamate you have in your media. Here in red is the suppressive effect, again on a log scale, of treating these cells with a glutaminase inhibitor if you have physiological amounts of glutamate, such as 1 micromolar. But if you have industrial amounts of glutamate shown in green, you get the published effect, which is in a log scale quite modest, and that's why they showed it on a linear scale. So rounding this out, so glutamate is the source in the cell for another very important chemical called glutathione. And glutathione is probably the most important chemical in the cell in terms of protecting cells from redox stress. And so this has been studied for years by cancer biologists because glutathione plays a very important role in sensitivity to radiation as well as sensitivity to conventional chemotherapy. And so Sam looked at his tumors, once again, that were either wild type for IDH or mutant for IDH, and he measured glutathione, which scientists refer to with these two different acronyms. In both cases, the glutathione was decreased, and in fact, others had already started to report that in real patients with IDH mutant gliomas, glutathione levels are low compared to tumors that have the normal version of IDH. So this suggested that IDH mutant tumors, in addition to lacking enough glutamate, are also lacking glutathione. And again, glutathione is a very important defense mechanism for radiation as well as for chemotherapy. And so we were very gratified to see this paper where these investigators reported that, somewhat paradoxically, having an IDH mutation increased your sensitivity to radiation. But now we understand the mechanism. It's because if you have an IDH mutation, you're being starved of glutathione, which again is an important protection against radiation damage. Sam could also model this in cell culture. I apologize for the busy slide, but in red are the cells that have IDH mutations, and in blue are the matching cells that have the normal version of IDH1. And then on the y-axis, he's measuring cell death as he adds in chemicals that are sort of radiation mimetics. They induce a lot of redox stress. And so you can see that the cells in red are very highly sensitive to chemicals or drugs that induce redox stress. And we know that this is specific because on the right, what he's done is he's given the cells a little bit more glutathione, and he can reverse the effects of the radiation mimicking chemicals. Lastly, we would expect that this would mean that an IDH mutation would sensitize you to radiation therapy, and there are a variety of ways of doing this, both in animals but also on plastic dishes. So here's a plastic dish type of experiment where you're looking at cell survival after cells have been treated with radiation, again on a log scale, not a linear scale. And in green are the cells where he treated with the glutaminase inhibitor and where the cells also had the IDH mutation. So once again, we're pulling the rug out from underneath these cells. They have the IDH mutation that's inhibiting the BCATs, and then we're blocking glutaminase. So now they're really struggling to make glutathione because they're depleted of glutamate. And then finally, if you grow cells in the brains of immunocompromised mice, as was done on the left, here on the left, the tumor has an IDH mutation, and in red, I'm showing you that if you just give them a few doses of radiation and the glutaminase inhibitor, you can see very marked improvement. And I should point out this is a very aggressive form of brain tumor, at least in a mouse model. And so even these effects are considered quite significant by our colleagues in neuro-oncology. And this is specific because if you do the same experiment with identical tumors that now lack an IDH mutation, you don't see any benefit of the radiation and the glutaminase inhibitor. And so we think this tells us that maybe one approach in the IDH mutant brain tumors and perhaps other IDH mutant tumors would be to consider exploiting this sensitivity to radiation and to radiomimetic chemicals. Now finally, there's one other way that we've tried to look for vulnerabilities, but you'll see in a minute that they kind of circle back. So one approach was to be completely agnostic. So, we had made some good guesses a moment ago, and we came onto BCAT1 and BCAT2, but let's just be totally agnostic. Let's just have matched cells that do or do not have an IDH mutation, and let's just start throwing chemicals at them and look for chemicals that kill the IDH mutant cells but don't kill the IDH normal cells. Maybe that will teach us something. So Sam and Diana decided to do this, and they collaborated with Joan Brugge's lab at Harvard and Isaac Harris and Jennifer Endres. So again, we have matched cell lines that do or don't have an IDH mutation. We just started with a highly selected collection of chemicals, about 500 chemicals, all of which have known targets, known functions. And then we're going to treat our matched cells at 10 different concentrations of the chemicals. And we're going to look for examples where the chemical kills the IDH altered or mutant cells, but is much less active against the IDH normal or wild-type cells if you look, for example, at cell number as a function of compound concentration. And we have very fancy and I'll point out fairly expensive machines for monitoring cell count in these experiments in a very quantitative way. So here's what the actual raw data would look like, at least to a scientist. So if a chemical doesn't discriminate between IDH1 and IDH wild-type of mutant cells, it's going to be around 0. If it preferentially kills the IDH mutant cells, it's going to be in the red box over here. The opposite tail is the opposite effect. So we're going to now focus on chemicals that seem to preferentially kill the IDH1 mutant cells compared to the matched IDH1 wild-type cells. So here's a list which is unreadable, I apologize, of the top scoring chemicals that preferentially killed the IDH mutant cells. But what was somewhat gratifying is when we looked at this set of chemicals, we immediately rediscovered some recent truths that have been reported in the literature in terms of susceptibilities that IDH mutant cells display. Just so as an example, it had been shown by my colleague Dan Cahill that IDH mutant cells are very sensitive to so-called NAMPT inhibitors, and here's a NAMPT inhibitor in the top 10. Others had reported a sensitivity to PARP inhibitors, and here's a PARP inhibitor, et cetera, et cetera. So that sort of validated the assay in hindsight. Now, it turned out many of the top hits were involved in pyrimidine synthesis, which again, I apologize for the jargon, but pyrimidines are building blocks for making DNA, a particular type of building block required to make DNA. And it turned out that multiple drugs that block pyrimidine synthesis preferentially killed or suppressed the IDH mutant cells versus the wild-type cells. So shown in red is cell number in the IDH1 mutant cells treated with, for example, this compound here, compared to blue, the matched cell lines that had the normal version of IDH1. So in every case, we could see a so-called therapeutic window where the chemical was more active against the mutant cells. This just validated in a somewhat complementary assay where here we're going to measure cell death induced by the chemicals, and in every case we see more cell death in the IDH mutant cells compared to the IDH1 wild-type cells. So what's the mechanism? Why are IDH mutant cells so sensitive to inhibitors of pyrimidine synthesis? And I should point out before I forget, one of the unfortunate truths in biology, including cancer biology, is context matters. So what I'm telling you is in our models of brain tumor, when Nabil and I were just discussing, I think it's too early to say whether this will or will not play out in the context of a cholangiocarcinoma, but I think it's certainly worth testing. So what's the mechanism? So the mechanism, we think, is as follows. So it turns out it has to do with BCAT inhibition, at least in part, because I told you BCAT makes glutamate, but glutamate is needed by the cell to make aspartate, and aspartate then gets funneled into the pathway that cells use to make pyrimidines. And so here, for example, 3 of our hits from our screen, and they're all acting in the pyrimidine synthesis pathway. So you can imagine why, if you're depleted of aspartate, you might be having troubles making pyrimidines. And so, again, I showed you these sort of fancy isotopic flux studies where now Sam has given cells isotopic labeled versions of the branched-chain amino acids, and he's going to follow their conversion into glutamate, aspartate, and now this Carbamoyl-aspartate. And in red, you're seeing that in each case, if the cell has an IDH mutation, the flux through this pathway is diminished. The cell's having trouble making this building block for pyrimidine synthesis. And likewise, if you go back and looked at cells treated with that cell membrane permeable version of R2HG, everything downstream of this point goes down in the IDH mutant cells, and you build up the precursors up here. So we know very clearly that this is where the block is in these cells. And then the last thing to try to understand is, okay, there are 2 types of building blocks for DNA, pyrimidines and purines, and it turns out that purine synthesis also requires aspartate, but Sam's a very good biochemist, and he realized that although you need aspartate for both of these pathways, the critical enzyme for pyrimidine synthesis, shown over here that you can't read, it binds to aspartate very poorly, and as a result, it's exquisitely sensitive to decreases in intracellular aspartate, whereas the corresponding enzyme that's used for purine synthesis has a very high affinity for aspartate, so it always wins compared to the enzyme that's trying to do pyrimidine synthesis. So at this point, we're trying to test some of these concepts in animal models to see whether, at least for brain tumors, blocking pyrimidine synthesis would be a useful approach, and then Nabil and I and others are talking about whether this can be transplanted to other tumors such as cholangiocarcinoma. So thank you.
Thank you very much, Dr. Kaelin. That was a beautiful talk, and we'll be saving questions for the end for the panel conversation.